Hi guys, this is Gaurav Chaudhary and welcome back to our online classes. Biotra and Learn. Please share and subscribe the videos and like our videos to make us inspired to make some more videos, right? So let's move on. Today we'll discuss about the genetic code and how does it come from. In the last classes we have studied about replication and transcription. Now let's move on to our next section, that will be the translation. But before that one, we have to know certain things about the genetic code and how does it develop. Now during transcription, what we have seen that and particular RNA polymerase moves in this particular direction and produces a particular mRNA, and this mRNA is coded by the actual gene sequence or base sequence what we have seen earlier, right? Suppose what's happening in this particular case, how these free NTPs or nucleotides, how these free NTPs nucleotides will join in this section. We'll see this one. So in case of transcription, we have seen that it produces a particular mRNA, and then rightwards what we have certain NTPs which bind to this section to produce that particular mRNA. So how does that particular NTP, how these NTPs will sit in against a particular base in this section. Okay, so let's move on, and while moving on to our genetic code section, we'll understand what is particularly a genetic code. Suppose what we have seen, suppose in case of numbers we use A, B, C instead of 1, 2 and 3. Now if I want to write CBA, then what would be the general code? The code will be 3, 2 and 1 in this section. So likewise, in case of a particular DNA, when it is transforming to a particular RNA, it will follow a certain code by which the further processing will be carried out. Okay, so suppose now we have A codon and G. Suppose these are particular codons, right? So in these codons, these things like G, C, A, T, in these particular cases, if we take a particular sequence of a gene, suppose this one or maybe this one, these 1, 2, 3, 1, 2 and 3 will be known as a codon. So how does this sit along during the process of transcription? During the process of transcription, suppose there is a code of G, C, A and T. So we are transcribing this particular section G, C, A. What will we get against this one? This will be C, G and T. So this is a kind of example that doesn't have any relation with the 64 codons we will be dealing with. So this will be the particular sequence. Now in case of transcription, suppose there are loads and loads of these kind of codons. So first we want to transcribe this particular section. So to transcribe this particular section, what we have to what we'll get? We will get that if C, G, T, C, A are certain NTPs, these are known as NTPs. these entities will come forward and sit in each place where they are required according to the code written in this particular section of a gene, right? So these will, suppose I have written C in here. So where will the sit C? C sit. C will sit in this particular section. I have written G. Then G will sit particularly in this section. I have written U. Then U will be sitting particular in this section, right? In case of mRNA formation. Lastly, what I have told you earlier, that this was T, but the T is always replaced during mRNA transformation by uracil. Thymine is always replaced by uracil during the transcription process to produce mRNA. Otherwise, the no translational work will be followed by, right? So this was the basics of genetic code. So these three things will be known as particularly a codon, and this codon will help us to transcribe a particular mRNA, and that particular mRNA will lead us to a particular protein structure, right? So let's see how it works and what are the different salient features of the genetic code. So in case of particular genetic code, that we know that there are 20 essential amino acids, right? Now for each amino acid there has to be a triplet codon. So there has to be a triplet codon. So each will consist of a triplet codon, right? So what we have in here, we can have n to the power q. N means the number of bases we find actually in a particular DNA or in a particular RNA. So in case of mRNA, in a particular mRNA, we get this number n is equal to 4, hence it becomes 4 cubes. Now 4 cubes will be 4 into 4 into 4, so there has to be 64 codons, and these 64 codons will give us a particular genetic code to translate a particular protein, right? So let's move on to see how these codon works and look at the salient features of it. Now, according to the bases and their combinations, there should be 20 amino acids, and according to the physicist George Gamow was the first person to say that there are four alternative bases or 4 NTPs which will lead to the production of these 20 amino acids, and these 20 amino acids will give us these 64 codons. Now what happens, there could be multiple codons for a single amino acid, so it will lead to a code degeneracy, right? But in certain cases there is ambiguity. So let's take a look, how does a particular codon look like. Okay, so suppose I am writing a code in this section. Okay, suppose I have written AUG UUU, then UUC UCU, then UUU. So in this section we can see that there are three pairs of bases in which we can find actually different kinds of codon. Okay, let's make it U. Right. So these will be our codons, and against these codons we will get particular kind of amino acid. Right. So what would be that one? So in this particular sequence, suppose this one, this will start a particular transcription. That's why it will be known as the start codon, or another name is initiator codon. And these are the functional codons. These are functional codons. These functional codon will prepare certain amount of amino acid and will help to place the charged amino acid, charged mRNA, T-RNA in this sequence. And how will it work? Suppose this is initiator and this is functional codon. I have written it earlier. So suppose this is methionine, right? This is methionine. For this one, this is phenylalanine, this is phenylalanine, this is phenylalanine, and so on, right? So what we will get? We will get M, P, 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 P. So we will get this kind of polypeptide sequence. This kind of polypeptide sequence from this particular codon, right? So let's move on to the next sequence. So in this case we can see that it is said that the first salient features of these particular codons is that the codon is a triplet and 61 codons code for a particular amino acid and 3 codons do not code for any amino acid. They are just the stop codon or the terminator codon. So what kind of functions they have? We have 64 codons. Now out of these 64 codons, 60 codons will generate a particular amino acid. One codon will work as a start codon out of this one, okay, and 3 codon will work as a stop codon. Now let's take this start codon, that is the AUG, and in case of AUG we can say that this AUG also codes for methionine. Hence it's coding something, it's charging the methionine and putting it on a particular place. So we can say that this one is also a amino acid producing codon. Hence total of 61 codons will code for a particular amino acid, and only a single codon will work as a start codon. This will work as a start codon, and these 3 codon will work as a stop codon. I will come on to that part later. So let's go for our next characteristic. There in this particular section it is mentioned that one codon codes for only one amino acid, hence it is specific. So what does it mean? Suppose we are having UUU or UUC. Both of these codon will specifically, whenever we get a code of UUC and then UUA, we will get a combination of leucine and afterwards phenylalanine, right? So these particular codons will produce these amino acid, but will place these amino acid, but there is a specificity. Hence, if you change this one, such as CUU, you will get lysine, leucine, right? Whenever we change certain thing, like suppose in this particular section we have that UUC. UUC, in this section we have phenylalanine, and then in this in the next one, if we change it to CUU, then it will code for leucine. So every codon, depending on the sequence of these bases, will have a particular amino acid. A particular amino acid sequence will be there, right? So this thing, whole thing is known as specificity. This thing is known as specificity. Now let's move on to the code degeneracy. Let's see code degeneracy. What is code degeneracy? Right. So what is degeneracy? In code degeneracy, suppose we are having UUU as phenylalanine, and next one UUC as phenylalanine, and another one we can have in this section that it will produce that more than one codon. In this section we can see that there are two codons in this section which are producing phenylalanine, and what we will get in this section, two different kinds of codons can produce the same kind of amino acid, but multiple codon is there. So we can have same kind of amino acid from multiple codons. This process is known as the code degeneracy. And now let's come to the stop codon. There I have told you that there are three kinds of stop codons in here. So in a particular process, the translation has to stop somewhere, right? Suppose this is my mRNA strand, and in this section I have UAA, or there might be UAG or UGA. All these codons will work as a stop codon. While when the ribosome, particularly when the ribosome come to this section, the different parts of this ribosome will be released, and the polypeptide it has produced earlier, that polypeptide will also be released from this section. So these three codons, UAA, UAG and UGA, are the potential stopper or the potential terminator of the translation process, right? So let's move on to the process of translation. Before that, we have certain examples of universal codon. So what is a universal codon? Let's see. The codon is universal. For example, for bacteria to human, we can say that UUU would produce phenylalanine. In certain exemptions are there. In case of mitochondrial codon, we can find different kind of codons in case of phenylalanine, right? So why codons are known as the universal codons. Codons are known as universal. Codon is universal. What does it mean? Codon is universal. What does it mean? It means, suppose UUU codes phenylalanine for human, and human means eukaryotes, right? Eukaryotes, and UUU phenylalanine for bacterium also. But this particular codon UUU, in case of mitochondrial DNA, mitochondria, we will find that it will produce a different kind of protein, a different kind of amino acid sequence that will lead to an exception in this story, right? So codons are universal. Why? Because in case of both eukaryote and prokaryotes, they produce same kind of amino acids. That's why they are called universal codons, right? Okay, so let's move on to our next section. Now guys, let's move to the mutations in case of genetic code. We'll study this one later, but now we have a requirement according to the chapter. So what are mutations? Mutation means a change in a particular DNA sequence. Suppose A, T, G, A, T, G, C, A. That's a sequence in this case. Now, due to certain reasons, suppose due to enzyme activity, number 2, heat, number 3, UV exposure, certain types of bases gets deleted. Deleted means that particular base vanishes out from this sequence. So the resultant sequence will have A, T, C, A. Hence, before we were getting a code ATG, but now what we will get? We will get a code ATC, which will change the particular sequence. Now let's look at it from a different angle with a proper genetic code, right? Okay. Now let's see. Suppose UUUC is a particular code in this case, right? So in this particular case, we can have that UUU, this one was coding for phenylalanine. Now due to certain reason, this particular UUU will show UUC codon, that is, this U has been deleted. So the amino acid in this particular section will be changed. Now in case of, suppose we have a different codon, like suppose let's see, alternation, right? Or maybe we can have a codon. Now let's see what kind of mutation can be done by the deletion process, right? So we are going here through mutations. these mutations will show that, suppose this was my first codon and this was GUU, and this particular section was producing valine, right? So GUU codes for valine. Now due to certain reasons such as exposure, this particular D, suppose UV exposure is there, this particular G is being removed, right? This particular, due to this particular removal, we will get the remaining portion UUU, hence no G will be present there, right? Now UUU will code for phenylalanine. So from valine to phenylalanine, we will get a different kind of amino acid, hence the protein structure will change itself. Now there is another thing that is known as the frame shift mutation. So let's see what is it. Suppose in case of frame shift mutation, now we have a code for GUUC. And that is our particular code, and this particular code sequence, actually what happens, this particular code sequence codes for valine, right? So in this particular case we can see that certain number of bases such as this one is being removed, and this G sits in this particular place. UU has been removed and G is been shifted. Shifted to this particular section. So what happens? Suppose if I delete this one, suppose GUUC was there, and now due to certain reason a pair of UU base is being removed, right? Suppose due to UV exposure, this base is being removed. What would happen? It will form GUC, and that particular GUC will produce another kind of amino acid, right? So GUC, basically what happens in this case, GUC produces a different kind of amino acid that is also valine. In this kind of case, the amino acids do not change that much. Previously it was also showing that GUU. So valine was from GUU and valine is from GUC. Hence no conformational change in protein takes place. In this particular section, in a particular protein, if it was valine, this is also valine, so no conformation will be changed, right? Now in case of shifting, GUUC was there. If these pair of bases are being removed, then what we will get, the overall frame will shift from this section to this particular section. Now it will UUC, it will produce phenylalanine rather than producing valine. I hope you have understood, right? So the overall frame shifts from one place to another. Now let's have a easier example. Suppose A, B, C, D, E. This was the previous frame. Due to deletion of C, we will get A, B, D, E. Hence, the overall frame shifted from this position to the next one. This includes deletion, and this is known as a frame shift mutation. Now let's see what happens in case of insertion. Insertion mutation. What happens in case of insertion? We will have a, suppose there is G, U, U, C, right? Previously, suppose U, U, C was there. Now, GTP was around this section and it is being inserted in this section. So first, what was happening? UUC was coding for phenylalanine, but due to insertion of G, that particular G is inserted in this section, this frame will start to produce valine. So what happens? A particular GTP is inserted in this section. This will be known as the insertion mutation. It will result in a conformational change in the protein structure. Okay, so we will be moving to our next topic, the structure of tRNA. Now in this particular section we can see there is a structure of a codon and particularly a gene sequence, right? So what is basically a tRNA? We know that there are mRNAs that we get due to the transcription in this section. Then we get rRNA or ribosomal RNA that helps in the formation of ribosome, and tRNA, which is known as the transfer RNA, that helps in the adapter molecule formation. Now what is the adapter molecule? Adapter molecule means, suppose we have a code in mRNA like this one, right? AGU. So it will contain a particular codon which will be next or opposite towards the anticodon loop. This is known as the anticodon loop. And it will help to bring this particular amino acid in the structure. In case of USC, we will get AUU, right? AUG. So AUG will bring tyrosine in this section, and all of these will undergo the peptide formation or peptidation reaction, right? So they will start to form protein. In the translation, we will get this mRNA being translated into protein structure. So the amino acid along with the tRNA and mRNA is known as the adapter molecule, right? Okay, so let's move on.